sessions. I think we had really good posters, and thank you all for submitting and coming today. So now I'm happy and very pleased to announce Lotte Meteyard, our special guest from University of Reading.
Oh, special guest.
Yes, special. Oh, you are all special guests. So Lotte finished her PhD at UCL, working on embodiment and semantic processing, and then completed her postdoc at the University of Cambridge with Professor Peterson. She is also a speech and language therapist, and currently she is at the University of Reading in the Department of Psychology and Clinical and Language Sciences. She's interested in many things, and including semantic processing, how language interacts with non-linguistic processes as well, and also the assessment and intervention of speech and language disorders. So she has experience working with different methodologies like behavioral stuff, fMRI, TMS, right, and patient groups, patient studies. And today she will be sharing with us her knowledge about a very, according to me, a very hot topic in language research. Statistics. It doesn't sound hot, but I think it is, because she's going to talk about mixed and general additive models. I'm sure you've come across those additive models when we read papers, new articles in journals. You've done it yourselves. We all try to do them. So Lotte is going to talk about those and how they might change our perspective about experimental design as well. So thank you for coming.
Thank you for that introduction, Izzy. That was just lovely. So with Guillaume's talk, he said it was probably the longest title that he'd ever had on the slide. This might be one of the most boring that I've ever had to give for a presentation, but I'm hoping not boring in content. I should probably say from the outset that I am still in the early stages of using these analyses myself. So I am not a statistician, I'm not an expert in mixed and general additive models, but I do think that they are extremely useful and very interesting. And you're going to get a little bit of a sermon today on bad statistical practice in psychology. It's something I feel very passionately about. So I think sometimes we can forget that actually statistical analysis is one of our primary methods when we're doing research. It's great to have lots of nice toys and to set up a really fancy experiment. If you don't have a way of analysing your data, there's no point in having collected the data, right? So actually our statistical analysis process really needs to be built into our empirical design right from the very beginning. So you shouldn't ignore it, even if you would really love to, and you certainly shouldn't torture it. And psychology as a field is pretty bad. And there's been lots of papers quite recently and lots of discussion about how we can make it better, how we can do better. And the message that I would like to share with you is that if you find it in your heart to love statistics, it will love you back. That's my experience. So psychology is pretty naughty, actually, a lot of the time. If you just go onto Google and you do a search, false positives and psychology, or whatever, you'll come up with a number of papers. This is just one example that I pulled off from Psych Science. We've got things like double dipping, both in behavioural data and neuroscientific data. So this is essentially the practice of analysing the same data set twice, going in and finding something that looks interesting, pulling that out and analysing it again, and surprise, surprise, it's still interesting when you analyse it the second time. fMRI has had a pretty bad time of it with not correcting for many, many different comparisons, to the point where people have analysed the dead salmon. This is a great sort of funny little thing, but it's an important point. Again, this is a kind of multiple comparisons problems. Because we use this p-value, this magical p is less than 0.05, it means that 1 in 20 of your comparisons are potentially false positives. That's just what happens when you use that cutoff. So you need to be careful about how many comparisons you're doing and correcting for that risk. And then more recently, again, there was a series of papers in Psych Science about how we have this obsession with the P.05 and how that means that we kind of have this undisclosed flexibility in how we analyse our data. So when we publish papers, we present the analysis process as if it was very straightforward. I collected my data, I dealt with my missing variables, I analysed it, and there I have my lovely significant result. But actually, the truth of what happens is more involved than that, and involves more kind of data manipulation, if you like. And there's a number of reasons why we're all pressured to do this, and that's a whole other discussion. But certainly my early experience of doing science and kind of collecting data and experiments was kind of spending a long time getting an item set together for a particular experiment, collecting the data, and then this was my kind of analysis process, right? So playing with cutoffs, cleaning my data, dealing with missing variables, averaging across individual items in a study to get my subject averages, that kind of thing, sticking it into SPSS and pressing go. If step 5 doesn't work, repeat steps 3 and 4 until it does, right? And then my feeling really after a while of this was, is this really what science is about? Because if it is, I can't be bothered. It's like, it just feels wrong. It feels wrong. It feels dirty. One of the reasons I actually went into neuropsychological research is because patients just produce behaviour, and you really can't push it around in the same way. And that's one of the reasons that I like working with patients. Or I see if they kind of face that. Or I try hard just to respect what you get in the first place and not push it around too much. Okay. So I'll talk about 2 kind of specific issues. That sort of browbeating done with. We have some kind of particular issues that in language research especially. So one is, say we're comparing 2 different groups of items, nouns and verbs, for example. How do we know that what we're getting is a real effect of that comparison between nouns and verbs, and it's not just confounded with other differences between those items that we haven't controlled for? And then also in language, actually, we're dealing with one of the possibly the most complex informational system that human beings have to learn and use, right? Language is very complicated. There's many different variables, there's individual differences, all this kind of stuff going on. And actually what we really want to do is understand and get a handle on that kind of complexity. Up front, I'll say that all the analyses that I'm going to kind of show you today have been completed in R. If you don't know what R is, you should all go and find out and retrain yourselves to use it. It is seriously the best thing I have done for myself in the last 10 years. I'm not joking. It's quite a steep learning curve, but then the payoff is huge. Right? Like, really. If you want me to, get on about how great R is, I can. It's fantastic. There's a great support community. It's free to download. It has something like 35,000 different kind of statistical packages that people have built for you to use. It has fantastic online support. It changes the way that you kind of think about your data and you do your analysis. It's just fab. So, I think these 2 issues kind of come up quite a lot in language research, so they're kind of pertinent things that we want to be kind of dealing with. The first one that I'll talk about is this comparing groups of items, and actually I think this is one of the reasons why mixed models kind of became a thing that we should all use. So this is a problem particularly when you have separate groups of items, nouns and verbs. It could apply actually to any categorical comparison that you're wanting to make. So actually it applies outside language research. It also applies to places and houses, like whatever. If you're interested in the difference between 2 categories, then this is a problem that you need to deal with. So these are situations where you have items nested in conditions. So separate groups of items. You've got one set of 10 words that are your nouns, a different set of 10 words that are your verbs. So your items are nested in conditions. So the question is, how do you know if I see a difference between these 2 conditions? How do I know that what I'm seeing is a noun-verb thing, or is it actually just some other confound between those groups? There's a couple of different ways that this can arise. One is that actually you just haven't matched on something. You know, we're only human, and there are as many different linguistic variables you can potentially match on as, you know, sometimes it feels like days in the year. The other is that you could have, this is an extreme example of an extreme item, but you could have a situation where actually you've got 5 items, so I've got 5 nouns, I've got 5 verbs, and what you can see is that actually in the noun and in the verb case I've got some items that are, one item, a few items that are just different. Okay? So you could have something in your item set where you've tried to control for everything, but for some reason there's a few of your items that are going to drive an effect that actually isn't due to the group, it's just due to that, those few items being weird for some reason and causing everyone to respond in a certain way. Okay? Just like you could have a subject who's weird, okay, and drive a kind of effect that perhaps isn't there across the group. The bigger issue with, I think, the way that traditionally language research has been done, although it's changing now, and I'm actually kind of hoping that a lot of what I'm going to say today is old news for everyone, is that we care about generalisation. So that's why we care about this at all, actually, is because we want to be able to generalise from our sample to the population. So at this point I'll introduce fixed effects and random effects. So if we do a fixed effects analysis, we're assuming that the effect size that we see is invariant. It happened in my experiment, if I did another experiment it would be the same. Okay? If you do a random effects analysis, you're assuming that actually the effect size that you see varies from one study to the next, and essentially your study represents a sample, a random sample from a population. Well, okay, what does that mean really? It means that if you know that something is sampled from a population, it should be treated as a random effect. So we do this for people, and we should also do it for items, because we know that our set of items are a sample of a wider set. In most cases, I guess you could say that closed class items are not accurate. That's an interesting point, just an interesting question. But if I want to say something about nouns and verbs, I want to say something about all nouns and verbs. I don't want to just say something about my 10 nouns that I used in my experiment. So this is a problem that was actually first raised in 1973. What year is it now? Right? So, before I was born. Great paper by Herb Clark where he said, look, we're all treating items as if they're not sampled from a larger population of items. This is mad. We need to sort this out. And unfortunately, his solution at the time was an extremely conservative analysis using something called min-f, f prime and min-f. So it was very conservative, so everyone decided that they weren't going to do it, because basically you hardly ever get a significant result if you do this min-f analysis. There were a bunch of papers that came after and that are more recent where individuals, particularly Rymakis, I never know how to say that name, looked at this problem and suggested some other possible solutions. Okay, maybe you don't have to be super conservative, maybe you only have to be conservative in certain situations, like when items are nested within conditions. People came up with decision trees for deciding when you needed to control for this and when you didn't. And actually, what's even more interesting is that what happened was that for a long time, language research reported separate subject and item analyses. So we would average over our items for each subject and do the comparison, and then we would average over subjects for each item and we would do the comparison, and we would report those separately. And we would say, yes, I have an effect by subjects, maybe I have an effect by items. If I don't, I'm just going to sort of ignore it. If you go back and read these papers, at no point does anyone say this is what you should be doing. This is just completely wrong. You've got just as much chance of getting a false positive in your subject analysis as you have in your items analysis, and presenting them separately gives you basically double the chance of finding a false positive effect. It's absolutely fascinating that this is what happened when actually no one said that that's what we should do. Right. So it's just a bit of history, it's quite interesting. Part of the reason is kind of the computational complexity of the sorts of analyses that that could be done, and that's something that has been tackled, particularly in the last few years, with the increase in computational power that we have available to us and packages like R. So yes, in my postdoc, when I did this reading, I was like, oh my god, I've totally done that in papers, and it's wrong. Okay, so items and subjects need to be treated as random effects, really so that you can say what's true for my sampled items is true for a population of items, what's true for my sampled people is also true for the population. And also because I think that we work really hard to match item sets on everything, and it was a point that was raised earlier about, actually, if you're using small item sets, you're ending up with underpowered research, and that's not good either. And so actually, we want to be able to work with large item sets, but then it becomes difficult to control for all the things that we know that we need to control for. So you're in a bit of between a rock and a hard place, really. But there is now a relatively simple solution which is computationally possible, and these are mixed effects models. Essentially, this is just returning to a more explicit regression procedure. So mixed models and general additive models that I'll show you later, they're just forms of regression. So they're not, I've heard people talk about things like structural equation modelling and things. I have no idea what that is. A mixed model is a regression, but it's a regression in which you tell the model what is fixed and what is random. That's basically it. A really nice paper, if you haven't read it, is one by Mark Brisboe, which you can just download, and it's showing you how to implement mixed effect models in SPSS. And it's also a really nice introduction to mixed effect models. It's really lovely. He's done everyone a massive favour by writing that. So I just show you an example. In my original plan for this talk, I was actually going to get R on my computer and show you some stuff in R, but I'm not going to do that. But if anyone afterwards would like me to do that, I can sort of show you how it's sort of implemented in the software package. So this is just an example from queuing. So this actually isn't an experiment in which items are nested in conditions, but I've done this random effect for items and subjects anyway, so that I can generalize. So speech and language therapists, people like myself, we often provide cues during speech production tasks to individuals, particularly if they have aphasia, to improve word finding. So if I show someone this picture, I can give them a phonological cue, so a k. And then in the experiment that I'm going to show you, they were also given in a separate session the same pictures, but then they just had a tone, so just some auditory stimulus that wasn't a phonological cue, but speaking. So this is data that was collected by my colleague at Reading, Dr. Arkita Bose. So essentially the task is, what's the name of this picture? And then you've got two conditions, a phonological cue and a control cue, and that's counterbalanced. Sometimes you had this one first, sometimes you had, I think my battery's on. No, it's fine. This one first, so it was counterbalanced across session. This was done with 10 individuals with aphasia. They were mixed group, different types of aphasia. They named 175 pictures. This is actually the Philadelphia naming test, which is a widely used set of pictures. And the dependent variable is accuracy, so it's a 0 or 1, basically. Did they name it correctly or not? So the questions that we wanted to ask was, does it help to have a cue? Does the kind of item that they're naming matter for how accurate they are? So we looked at phonological complexity, the amount of semantic support the item had, and then we also had this variable, which was a kind of picture complexity name agreement variable. And then we can also ask the question, does the cue help for some items more than others? So looking at how cue interacts with these types of variables. this is a bit naughty to sort of present it as being so much more simple than the other one. But I think that when you start to work with these models and you think about the kinds of experiments that you're going to set up, what you actually think about is, okay, I need a large sample of items that represent the things I'm interested in, particularly if they're continuous variables in a good way, a good representation of all the things I'm interested in that set of items. Collect your data, then you model your data. And you find a model that fits your data and makes sense. So actually you fit a succession of these different regression models, particularly if you're working in R, and you find the best fit to your data. Okay? Obviously the models are motivated by the sorts of questions that you want to ask, but this model fitting is the analysis process. You're not throwing any data away, you're not averaging across any conditions, you're keeping it all, you're keeping all of your data and you're analysing it. Okay? Which I really like. And then, I think the really interesting thing with working particularly with R is that when you report what you've done, you actually report this model fitting process, and you can also share your data. You can just say, here's my paper, here's the series of models that I fitted, here's the final model that we think fits the data best, here's my data, here's my analysis script that I fitted the models. You can repeat that if you want, or you can do something different with my data if you want. So you open the whole thing up. And that's also really important for us to do, I think, if we're going to move forward, to share data and share these kinds of analysis processes. So here's an example of the kind of model you might fit. So we could have, for example, a model in which I've got the response, this is accuracy, and I want to know whether it's predicted by Q conditions. This is phonological Q versus control. Phonological, this is actually, these three things were the products of a principal components analysis where we chucked in a load of linguistic variables for these 175 pictures, and what we got was actually three quite nice components, which were, broadly speaking, this represents items that are kind of length, phonological neighbourhood, that kind of thing. Semantics, this was imageability, age of acquisition, and then this was picture complexity and name agreement for these of those. I would like a random effect for items, I would like a random effect for patients, and I'm going to stick in trial as well, just to see if that does anything interesting. So do the patients get worse over the course of the naming, do they stay the same? It's just some other sorts of variants that you might be interested in looking at. Or I could have a model where I want to know if there's an interaction between Q and those three item variables. R is very nice, or the package that you use in R, when you do this it will also look at this. So just because you've specified the interaction doesn't mean you don't get the individual main effects as well. So here's some data. These graphs are straight out of R, so I'm sorry I haven't had a chance to make them really pretty. So does the Q help? Yes. So here is phonological Q, no phonological Q. This is accuracy up the side. So you can see that overall, when you give people a phonological cue, they are more accurate in their naming than when you don't. That's very reassuring. We actually did the same experiment with semantic cue. That doesn't help at all. Does item type matter? Yes. So here what we have is, across the bottom, you've got these principal components that represent the kind of phonological complexity. So items which have a higher score on this component are longer, they have fewer neighbours. Where's Catherine at the back? She can't remember what else goes into these components. So lower frequency neighbours. So longer and less well supported, if you like, phonologically. So you can see that actually those items are less often named. Basically, they're not as accurate in naming those items. This across the bottom, that shows you the representation of my items across that component. So you can see I've got reasonably good, a bit sparse up here actually, but reasonably good representation of this component across the items. Here we've got this semantic components. This is age of acquisition and imageability. So items which were acquired earlier, more highly imageable, they're named more accurately. And again, you can see it's actually getting pretty sparse down here, so you wouldn't necessarily trust what's going on down here. But here, this seems pretty good. So, okay. And this is independent of Q. So Q doesn't interact with either of these. What Q does interact with, which was quite interesting, is this picture complexity, the name agreement component. So what we see here is your condition with no phonological cues. This is just the tone. Here is the condition with the phonological cue. And then you've got name agreement. So items that are score higher on this variable, they have, what is it? It is higher name agreement, more visually complex up here, lower name agreement, less visually complex down there, yes, that's right, sorry. And then accuracy again. So what you actually see is giving someone the initial sound helps them to name items where there's lower name agreement, helps them to name items where it's harder to find the name for that item. So kind of counterintuitive initially, because you think, oh, why isn't it making a difference for items that are like more phonologically complex? Why doesn't giving phonological information help you retrieve more phonologically complex words? And it's because of the nature of what a cue is doing. Actually, in this experiment, when you present a picture and then some phonological information in the cue, you're reducing the possible items, possible words it could be for the patient. And actually, this is interesting because most speech therapists, we assume, or like when you're trained, you assume that a phonological cue is supporting someone's phonological system. No, it's not. Or not in this task, anyway. So that's kind of interesting. Okay, so let's have a look at the random effects, because what are they doing? Here's a really crazy picture, but these are all the words, okay? 175 words. What I've done is I've ordered them according to their intercept, basically. So what the random effect does is it fits an intercept, a different intercept for each one of the words. If you imagine your regression graph, you can have an intercept at the bottom or further up or higher or lower. So we've got some words which are named much more accurately, some which are named much less accurately, and then a whole bunch in the middle. So you might, if you were interested in item variants, you might want to go and look at these items and have a think about why they're acting so strangely. You might want to use that as a way of not using these in further experiments. These items seem a bit strange for some reason, so when I do my next experiment, maybe I won't use them. Maybe I'll just take the ones that appear to be much more grouped together, because there's something about these items which makes these ones much easier and these ones much more difficult. So you're getting information from these random effects, which is nice. Here's the random intercepts for the patients. So here we've got, again, I've just ordered them according to, this is now kind of naming ability. So we've got a couple of patients here who are doing quite well, better than everyone else. One patient down here who's doing much, much worse. And actually we know about this patient that they've had 2 strokes rather than 1. So they're more impaired. Okay. So basically, using the models you can control for both subjects and items as being sampled from a population. That's very nice, it's statistically sensible. You can also get information about subject and item variance, and you can specify in these models whether you want to look at a continuous variable or whether you want to look at levels of a factor. They're really flexible, they're nice. Okay, I'm going to try and, this shouldn't be that long actually. Okay, so mixed models can help you to also understand, looking at many different variables, looking at individual differences and trial order effects. There's a guy called Rob Davis at Lancaster who's doing really nice stuff with mixed effect models and individual differences. He looks primarily at reading, but his stuff is really nice examples of using these mixed effect models. But what I'll finish with are these general additive models, which are kind of like another step again, if you like. So when we do regression, linear regression, we're basically fitting parametric lines. We're fitting lines which can be simply described by some function, if you like. But what if you have non-parametric effects? In other words, what if you have a wiggly line? Okay? Really problematic. Wiggly lines can pop up all over the place. Here's an example. This is pupillometry data, so this is the pupil diameter and how it changes according to cognitive load. We've got a multiplication task here, and you can see that, yes, you see differences in pupillometry according to the cognitive load of the task, but actually the fundamental shape of the pupil response is a wiggly line. So this is something that would be quite difficult to just model as a straight line, or with kind of standard parametric fittings. Wiggly lines can also pop up in every experiment. So, for example, the change in a participant's reaction time over the course of an experiment. People get tired, they slow down. People get bored, they speed up. Wouldn't it be nice if you could model that? Da, 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 da. So, general additive models, again, it's basically a regression procedure. However, it gives you the option of fitting wiggly lines. You can tell it to fit a straight line, if you want, or a wiggly line. The downsides, it's computationally pretty heavy, so your computer will sit there chugging away whilst it's trying to fit these models. It's also quite happy to fit the data. So you'll give it some data and it will go in and it will fit, and sometimes what it's done doesn't always make sense. So this process of model fitting, you actually need to go in, fit a model, graph everything, look at it, think whether that makes sense or whether actually you haven't got enough power in there for that to be fitted. So you have to be a bit more careful with it, I think. So here's just an example of one of these. This is a different task. So this is a property verification. So you're given two words and you basically have to say the second word, the physical part of the first, yes or no. So these are the items that we're interested in looking at. Conditions where the response is yes, monkey, leg, yes, leg is the physical part of the monkey. We have two different conditions in which these experimental items appear. So one where the filler items are related. Monkey, banana, no, a banana is not a part of the monkey, but they're associated. Or unrelated. So chair is definitely not part of the monkey. 30 subjects, 100 experimental trials. Dependent variable is reaction time. Okay. Got some variables of interest. I'll look at the effect of fillers, whether they're related or unrelated. I'm expecting people to be slower when there are these related fillers. It will just make everything a bit harder. I'll look at the association strength between the object and the part word. I'm expecting when that association is stronger for people to be faster. It can also chuck in frequency of the word, length, and so on, other things that you think might be relevant. Specify a model again. This time you fit what's called a smooth. It's a wiggly line. Fit me a wiggly line, basically, is what you're saying. But this time, I'm also going to fit a wiggly line for each subject over the trials. So I am going to model this variation in the subject's reaction times over the course of the experiment. So, sorry I haven't been able to have the time to convert this back into reaction times, but this is the effect of condition. So related fillers are harder, so here's related fillers, unrelated fillers. Overall, people are slower in that condition. It's just to show you that you can pull out, just simple comparisons from these kinds of models as well. Here's basically a linear effect of association strength. So when there's a higher association between the object and the part word, that's along the bottom here, this is reaction time, people are quicker. Okay, so again, quite sensible. I love this graph, I think this is great. So here we have our subjects, here's the trials across the bottom, number of trials. Each line is a subject, and then this, there you can see you've got, they're all, these people are really fast up here, this person is really slow, and then what you've got is their change over the course of the experiment. So here we've got someone basically getting bored and speeding up. Ah, I can't, where is that? Yeah, speeding up, no, so, I'm a little confused, it's late. Yeah, getting faster. So, yeah, I'm doing it, it's getting really boring, just going fast. And then you've got other examples where people are getting tired, so there's someone fatiguing and slowing down. But this is now variation in subject and trial that I've taken out of the model, and so it should leave room for my other things that I'm interested in to suck up variance that's left. So this is a way of, you might be interested in looking at changes over time in an experiment, in which case this would be interesting in and of itself, but you might also just want to model this to take it out as something that's going to be potentially confounding what you're interested in. So general additive models do the same kinds of things as mixed effect models, except you can fit these non-parametric effects. And again, you can specify whether variables are continuous or factors. So they're pretty good, they're flexible. So, of course, these aren't appropriate for all research designs. There are still situations when it's perfectly fine just to go and do a t-test. But consider modelling your data with variance left, especially when you've got a complex data set. And I think that the more that I get into this sort of stuff, I think that it becomes more important to go through this kind of explicit modelling process, because actually we want models of cognition and language. And I think when we start to think about things in this way, we can move a bit closer to that. So you need to be thinking about experimental designs that give you sufficient power and sufficient variation, particularly in your items, to support these kinds of models if you want to use them. So what kinds of designs will give you that? I've given you two simple examples, they've all been with single words, but they're not necessarily more onerous than your normal kind of experimental paradigm. And from the statisticians themselves, you're fitting the least worst model. Okay, that's what you're doing. So, just to reiterate, I think this modelling process, using all of your data and finding a model that fits the data best and reporting that model fitting process and sharing your data is a much more satisfying way to work. So I say thank you to all these people for helping me, and the British Academy for allowing me to spend time with Harold Bayen and learn R. And my new personal hero, he's a statistician, was a statistician, he's passed away. But yeah, very good to remember this. Okay, thank you very much. Is that alright? Did I run over?
No, you need no, just for 3 minutes. Okay. But we have time. We have time for questions, so please. Yes.
Do you encounter people who, when you try to get this kind of approach to doing statistics, and you, well, that's great, but it's not how everyone else does it, so you can't do it either? Because I think it's really
Wow. The short answer to that is no, but I am at the very beginning of my journey with using these models. So maybe that's going to happen. I mean, with any kind of analysis, it comes from there's a rationale for the analysis, right? And I think if the rationale for your analysis is strong, that argument is just irrelevant. So if these analyses actually will answer the questions that you're interested in, then it's okay to use them. And I think you have to feel that those are going to give you something more than what you do currently, I think, right? So if actually what you tend to do are simpler experimental setups where you're comparing things, perhaps using a standard ANOVA or a t-test or something, and that's appropriate for the design, then that's fine. But I think if you're interested in perhaps looking at frequency effects, imageability effects, things that you know are continuous variables, why even split them up into categories? It's a completely, if you think about it, it's a bit mad to do that. Why not actually respect the continuum in the first place, select items that vary along that continuum and then explore that variation? And I mentioned Rob Davies' work at Lancaster, and he's starting to do it to look at individual variation. So he's looking at age, reading ability, things like this. And so it allows you to get a handle on things that you otherwise might have thought were too complicated, maybe, and that you just needed to match for or kind of hide. But I don't know, it's an interesting question. I guess my other response to that question is, God, I really hope you don't get that response. Because that's doing science, come on.
Unfortunately, you do get that response. And different editors have different ideas of what's an appropriate way to analyse a data set or not. So I think what you did was beautiful and it was incredibly clear. And I am now enthused, although I have learned to hate R.
Oh no!
So maybe I need to pay you to sit beside me for a while.
I'll take cash. Cash is fine.
But it's quite frustrating if you have. I'm a good old-fashioned standard, theory, three things girl.
Yeah.
Fit it into an ANOVA
Yeah.
and see if it differs. When you try to publish this then you have to use these models in the narrative text.
Right.
It starts to get kind of irritating.
Yeah,
I
yeah.
don't see what it's telling me over and above
Yeah.
what I planned to
Yeah.
look for.
I guess that's quite interesting. I think there's two ways to sort of tackle that. One is that the models don't have to be complicated. So you could have a model that was literally a random effect for subjects, a random effect for items, and your factors. So it's literally the
But
original.
what is that telling me over and above my...
It's controlling for items as a random effect.
Okay, and the F1, F2s that we've all been doing for decades
Yeah.
are completely wrong then.
I know. Yeah, and it is. You should. Reading the papers was really fascinating. So I guess it's controlling for items as a random effect.
I liked my F2s. they told me things.
They did, but
they told me stuff that would make me go back and... Because I would always look at my data from every angle, so I'd actually know what I was expecting to come out of my analysis. You go into your analysis knowing what you should be expecting because you've looked at your data pattern in a variety of ways.
Yes.
But, F2s are hard to get, and if you're not getting them, it's telling you something about your material set.
Yes, and it might also be telling you that your F1 is not reliable.
Yeah, there's that too.
You're going to ignore it if it's not.
So the other
but you need them, you don't
the other thing I want to say is that because these are regression models, if you have a data set which actually hasn't got sufficient data points for a regression model, that's a very good argument for not doing a mixed effect model. Do you see what I mean? So actually you need to treat these like they are regression models, so you need sufficient data points and you need to follow the rules for using regression. So if you've got a situation where you've perhaps not got a huge number of items or a huge number of subjects, then in that case an ANOVA probably is more appropriate, because if you stick it in a regression model, it's going to be underpowered for the regression, and that could be a counter argument to applying a mixed effect model just because that's what we're all doing now. So there is a bit of a return to actually what are the statistics, and if you're doing a regression, then you need a good number of subjects, good number of items, good support across the things that you're interested in to support that kind of analysis. So yeah.
And bearing in mind that there is this objection that people aren't necessarily all using mixed effect models, therefore people don't necessarily understand them, what should we be reporting? Because I know you said ideally you should put all your output out there, but I've not read a single paper so far that's people all of that output in, given the description you do yourself. Some people just report t-values and p-values, some people write the slopes and the slopes. So
So there's a problem there with the journals and what they're asking people to report, right?
Well, I've not been asked to report anything.
You can make it available yourself. You can say, my data file is available for download on my website if you want. What tends to happen, and this is more about working in R than it is about using mixed models, is that you'll have something called an R file, which is basically a list of what you did. It's a list of things you typed in to make the analysis happen. And then you'll have your data that will be saved in a workspace file, and you can stick that and make that available for download so people can get it. So you can just do it yourself. As this stuff perhaps becomes more used, it might be that what's going to happen is we develop reporting procedures as we go. We find our way, I guess. And I think it requires you to probably be a bit bloody-minded about reporting things, and perhaps choosing to report more than what you might have been at. I mean, it's about not sweeping stuff under the carpet to a degree, and actually kind of just taking the data as it comes, if that makes sense.
Sure, yeah.
I mean, I don't have a straightforward answer to that because I'm not a journal editor and I don't, you know.
As a bare minimum, say, what you said, you have to present to make this kind of transparent.
Do you mean in reporting the results of a mixed effect model? I mean, then it's very much like reporting a regression.
Yeah.
So you would probably report. You might report your final model, but then you would describe the other things that you fit and didn't soak up significant variance, so weren't then included further.
And you tried to just say, we give this, I think it's
Our first models included interactions between X, Y, and Z, and we also fitted this, that, and the other, but those didn't, you know, contribute significantly to how well the model fit the data, so we didn't include them in the final model. You know, if you want to analyse my data set yourself, please email me and I'll send it to you. Yeah.
Yes, just one more.
As a journalist, I can tell you that the main one is that the heck of a lot of people don't understand what they're doing, and we are really quite worried about different. You know, I'm sure there are things being published quite regularly that are just not done. Like, I know there are legitimate controversies of this, like with your maximum or not, and you know all these kinds of things. So there are clearly different arguments about before you should do them. But then we also have the issue that I see with my own students when they run a different R script and suddenly their values, just their key values, just change totally, and it turns out that they've picked up some other piece of free software from the sort of R toolbox. It turns out that it's quite, it's making quite different assumptions. No one knows what those assumptions are.
Yeah, okay.
It really is a state of there's quite a worrying sort of state of chaos at the moment.
So there is a downside. The downside in using R is that people will design packages and make them available, and you can use those packages in a way that you can be a bit blind to some of the underlying assumptions. So I think that is probably an issue. However, you can implement mixed effect models in SPSS, and then it's very transparent what you've done, or kind of should be, if you like.
Yeah.
So that is perhaps one of the issues of using an open source software like R. In terms of people not knowing what they're doing, I don't think that's necessarily different to how things were before, statistically.
But it's very hard for reviewers to realise when the authors don't know what they're doing. I mean, this is the issue. Is you just genuinely don't know at the end of it whether this person's got it right or not. It's much harder than. I mean, I used to occasionally wonder if people couldn't do an F2. You occasionally got that. You just look. If you look carefully, you realise what they've done wrong. But you can't. At least maybe I'm not sophisticated.
I don't know if I agree, because, like I said, these are regression models. So I don't necessarily see it as being any more complex than reporting regressions.
Because they haven't been trained on that sort of problem. I mean,
yeah.
some half been trained on it.
You can do something in R and it spits out a result, but it actually left out your variables because there was something wrong with the data file. And
Yes.
You wouldn't know it. There's lots of things you can do in R.
Yes, and that's why data checking before you do your stuff in R is what you'll spend a lot of time doing. You need to go through a process of making sure that all your data is there and that R has coded it in a way that makes sense and that you want it to be. Again, having gone through the process and learning R, I disagree. It's different knowledge that you require.
How long does it take you to have?
I got a bit of the British Academy have a scheme called a skill acquisition grant, which is specifically for people in social sciences to acquire quantitative skills. I applied for one of these grants. I visited Harold 3 times. Probably in total I've spent about 2 weeks visiting his lab. I've been playing around with R since October.
Do you want to come and visit?
And there are people who are doing. It's different knowledge. It's a completely different way of analysing data. So whereas before what you might have done is get your data out of the computer, stick it into an Excel spreadsheet, do averaging and cleaning and stuff in Excel, take your carefully massaged data from Excel into SPSS and press go. all that stuff you're doing in Excel, where you're averaging and da-da-da-da, what happens in R is that you get your data out of the computer, you get it into R, you do all this checking and stuff in R to make sure that, yes, everything, R knows that this is a factor, R knows that this is a continuous variable, I don't seem to have any missing data, you know, da, 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 da. So that checking process goes on in R, and then when you're happy that the data set is complete and ready to go, then you put it through the models. But that data checking process it is the most time consuming thing, because the actual running of the models is like that. So it's a very different way, but when you've got to the end of that checking process, you know that all your data is there and it's all coded. You haven't done anything to it except check that it's in the right format really thoroughly. You haven't averaged anything, you haven't removed anything, you haven't you haven't done anything like that. So I sort of...
Do you never get rid of subjects?
Uh,
I mean, there must be. Yeah, the guy who came in and fell asleep.
well, I mean, again, I'm at the sort of beginning. So in that property verification, 2 of the participants aren't included because they didn't complete both sessions. So they didn't have a full data set. But now I'd be very tempted just to go for more power. Actually, don't throw that subject out. Just get a new one, or just get more, and just have more data and solve the problem that way, rather than removing data. That would be my approach now, I think. Yeah. So yeah, I kind of agree. We need to be more structured about how we're doing things, but we were doing stuff that was completely wrong in the first place. And personally, I think this is an improvement. So, yeah.
So let's